My name is Ling Cheng, and I'm an assistant professor here in the Faculty of Engineering in two departments, IBBME, and that's the Institute for Biomaterials and Biomedical Engineering, and also Electrical and Computer Engineering. I'm also a scientist here in the Ted Rogers Center for Heart Research. My research focus is on advancing medical imaging technologies, in particular magnetic resonance imaging, MRI, and the underlying motivation behind all the research projects that are going on in my lab right now are focused on early detection, very early detection of diseases spanning all the way from cancer to cardiovascular, and also technologies that would enable new therapies to go into the clinic. So in order to achieve both the early diagnostics and the enablers for these new therapies that are coming out, regenerative medicine, my lab focuses a lot on functional imaging and cellular and molecular imaging. CRISPR has been used to target specific sites within the genome sequence, sites preferably that are safe so that they don't affect the functionality of the other parts of the genome. Fluorescent tags can be put in, and those are used. One way to use them is to identify if your insert is actually integrated with the CRISPR technique. So you could have tags that are fluorescent, and what we're currently working on now in one of our projects is inserting MRI dyes through CRISPR technique into the genome, so that instead of looking at something on fluorescence microscopy, we can actually look deep in the body, deep in tissue, in a human, on MR to see if the genetic modification has been made. So there are multiple ways with which you can visualize how things are inserted. Fluorescence is one of them, MRI is another, and there are other techniques that you can consider as well. One issue is on scaling up. How do we actually scale up enough stem cells so that you can inject it into the diseased part of the organ and be able to regenerate tissue? I mean, growing enough cells to inject into a lab animal, a rat, is one thing, but in order to generate enough cells, we're talking about billions of these, into a human patient is another. And the cost of stem cell research is enormous. I mean, even working with stem cell injections for rats, we're talking about tens of thousands of dollars for a subgroup of animals. And when we're looking at pigs, so you could think they're sort of an equivalent to humans, $100K could probably get you enough materials and cells to do work on five or six pigs. So the costs are really prohibitive. There's another limitation that people are currently facing with stem cell treatment is that if you're injecting stem cells into an already harsh and diseased tissue environment, the chances of survival are not 100%. So how do you know that the stem cells that you're injecting are actually surviving that harsh environment and going on to become new tissue? Currently, we don't have any way of tracking how the cells do, how well they do, whether or not they survive, not days, but also weeks after cell injection. And then a third limitation of stem cell therapy currently is that it doesn't happen all the time, but sometimes there could be unintended differentiation. So you might want the stem cell to go in a certain direction, right? Differentiate, say, into a cardiac cell, and in order to do that, to make sure that the stem cell doesn't differentiate into non-cardiac cells, you actually do the differentiation outside of the body, and then you implant the already differentiated cardiomyocytes into the patient's heart. That has a limit because you're limiting the proliferation potential once it's already in the differentiated state. So ideally, you want to go back a bit higher up, but once you do, there's no guarantee that it's going to differentiate into this right cell type. I would say that because there's still unanswered questions as to the potential risks associated with gene editing, but you still need to demonstrate efficacy in human patients. So probably the best group to not be guinea pigs, but to be in a trial to test out these therapies would be those end-stage patients who really have no other option. And for them, this might be the one treatment that will save their life. My personal view on this is if the condition is something that is life-threatening, of course, there should be no question and no qualms about doing genetic editing. If the condition is something associated with a debilitating disease like Down syndrome or physical handicaps, I also feel that those people should also receive treatment. But if it's a matter of changing the hair color or changing the shape of your nose or your eye color, those are really, in my mind, really silly things to fret over because they're not debilitating and they're not life-threatening. So I think it's our job as scientists, once we get to a stage where we say, yeah, the results are very promising, we should really go out. Talk to high school students too. High school students are, they're the next generation of bright minds. Talk to them. Get involved in these extra, like one of my students gets involved in a lot of the science rendezvous where he gets science out to the general public and talk about our research. And I think if enough people are doing CRISPR but do that for their own part, right? The public would know about CRISPR just as every person that I meet in the dog park knows about MR now. Everybody knows about MR. We actually may be able to modify cells so that they are more responsive to therapy. So I'll take one example. So with breast cancers, a lot of the breast cancers that women have, many of them are not harmful. They could be treated with hormone therapy if the cancer's subtype is responsive to hormones, if there's responsive to estrogen, progesterone, and HER2, the whole bit. But there's a group of breast cancers called the triple-negative cancers that don't respond to anything, and those are the killers. Those are the ones where the survival rate is not very good. So if genome editing, I'm not sure how people could do this, but if genome editing can somehow edit our cells, and this would involve actually editing every single cell in the body, such that they would all respond in some way to hormone therapy. Sounds crazy. Then maybe we could get rid of these hormone non-responsive tumors. There's a lot of genome sequencing and even proteomics going on right now to identify specific markers for disease. But when you look at those, when you look at the analysis, it's crazy. A lot of genes could be implicated, but not every single gene will guarantee that cancer will develop. How many genes do you have to knock out? Right? That's what always goes on in my mind. Okay, you've identified all the genes. Do you knock all of them out? What's going to happen to the patient? So, modifying the gene so that it's not cancerous is one angle, but modifying your cells so that they respond to a certain type of therapy might be a more tenable approach. I don't believe in it. I don't. I think biology is too complicated to be simplified to an electrical circuit, say, with inductors and capacitors and resistors. Even complicated mathematical system like neural networks, which they now call artificial intelligence, it's hard to capture all the complexity of biology, right? Because biology is dynamic. Things can change depending on the environment. And when we look at a electrical engineering system or a mechanical system, it's fixed. How do you train that system to become dynamic with time? And it's also a stochastic process, biology. It's hard to predict how things are going to unfold. So my personal view is I think biology we should not try to simplify it to the point where we could model it in terms of, electrical engineering modules. For anything that involves light, it has to be shallow tissue penetration. And there are a number of things that could be done. If you could make sure that the light can penetrate at least a few millimeters, and that's deep, then you could start addressing problems related to melanomas. Colon cancer would be another. It's a big killer, and I would say that anywhere along the gastrointestinal tract, esophageal cancer, that's also pretty prevalent in Asian countries. That would be, it would benefit a lot of different types of cancers already. For a lot of the other solid tissue tumors, breast, it's hard. It's hard. Sure. Unless what you could do is you could introduce the light through an optical fiber. So that would be minimally, so now you're not relying on there to be like a gastrointestinal tract for you to insert your light source into, but you could use a fiber optic cable. It would be minimally invasive. I had one here, my student took it to sick kids. It's a thin fiber, make a small puncture, it's minimally invasive, and let's say you're targeting breast tumor, lung tumors, liver tumors. You could get that thin fibrotic probe to go in, turn on the light, and treat that way. So I would say be creative with your light source to make sure that you're able to access the broadest number of applications possible.